Next, we have Michael Chen. He is the CEO and co-founder of Great Bay Bio, which probably everyone knows by now because you can't miss the sign in the hallway. He has his PhD in biochemistry from the University of Georgia. He has more than 30 years of experience in the CMC, and we're all very excited to hear his talk. Thank you.
Thanks for the introduction. As you know, you might have heard some word called tech bio. That means we use a cutting-edge technology and data-driven technology apply into the biotech industry. So Great Bay Bio is a tech bio company. So we are using AI technology into the biotechnology platform, including CMC and drug discovery. So our goal is trying to make biotechnology faster and better. So why we want to use AI technology into the biotech? So in the biotech industry, normally any kind of a project, we are split into three steps. Number one, we have to use the human design to make decision what kind of a disease we need to cure, what kind of molecules, what kind of a target. So we have to use a lot of human design. Once we make a decision, we have to take a lot of time, a lot of efforts, a lot of work to find those molecules. Finally, in the step three, we make a confirmation with a clinical trial or animal trial. So our basic logic is of the Great Bay Bio is using AI to replace the second step, which take a lot of blind screening, a lot of repetitive work to get this done. So I think right now the AI technology in this stage can be done in the second step to replace a lot of repetitive work. So today, I want to show this. This is the whole value chain, starting from antigen development all the way to the IND filing, clinical trial, commercialization. So all those four red dots is our four platforms in the last 4.5 years to develop with AI technology. So as you can see, in the first red is we're using AI to do the antibody sequence generation, use the AI for the druggability and assessment. The third one is a cell line development, and the fourth one is culture development. So the first one, which is a major topic today, is how we use a site-specific integration platform in the cell line development. So we call AlphaCell is a site-specific integration technology. So as you know, in the traditional process, everybody, I think most of the audience are experts in the cell line development, is a random insertion. So your GOR will insert randomly in a different position. So they end up, these cell pools are ununiform. So we have to take a lot of time to trying to get the single colony is a high titer, then take another 3 months to get the stable cell lines. So it's a lot of work. But with our AlphaCell process, we take only 1.5 months to get a cell line. So what we do is we use the AI technology to find those hotspots in the genome, which is a high titer, which is stable. And every time the GOI express, we are transfected into the same place. So our cell pool is uniform. Once we have a uniform cell pool, we can do a lot of work. You need less work, less 96 well to get single cells. You don't need to necessarily to worry about the stability of the single cell because the site has been pre-selected as a single one. Also, in this cell pool stage, you can make some materials to evaluate your product, particularly for, we're going to talk about it later on for the biospecific. So from these slides, there's two things we need to think about. Number one, we can get a uniform cell pool. Number two, how we get this hotspot. So in a lot of research papers showing that the cell morphology, including cell size, nuclear size, the ratio, the number of organelles, is related to the cell titer. So that's some kind of the relationship. So we are using a lot of data, which is more than 100 million cell images with their corresponding titer make a AI training to get we call Clone 4.0 platforms. So this algorithm can figure out once you have an unknown cell image, they pretty much can tell you is a high titer or is low titer. This is a video. Here we go. Yeah, so we put the cell pool into the dish. Then this microscope, high throughput microscope, can take 150,000 cell images in 15 minutes. Then uploading to the server, we have an algorithm to figure out what is a high titer on the microscope and find the location. Then we have a robot go directly to that specific cells, take them out as a high titer single cell. So this is our basic in-house platform to pick those high titer platforms. So we're using this technology, as you can see, if it's an algorithm is a high accuracy or not. So this is 5 different experiments. Every dot represents 1 cell. So we use a GAP as a label to show in the titer. As you can see, those circled dot is predicted by the algorithm. As you can see, most of the predicted cells is in the high titer level. In other words, our algorithm is pretty much accurate to find those high performance cells. So the first advantage of this alpha cell platform is in the early stage, we can evaluate product, particularly for the bispecific antibody. As you know, in the bispecific antibody field, there's 2 headaches. One is a low titer. Of course, some of them may not have this kind of problem, but most of the time we have low titer headache. Second one is a low correct pairing, missing pairing percentage. So we can use this platform to solve these 2 headaches in the industry. So this is the summary of different kinds of the bispecific antibodies on our platform. Some is 2 chains, some is 3 chains, some is 4 chains. So as you can see from titer-wise, it's about 5 grams, 6 grams, sometimes it's 12 grams. So right now we can claim in the public that we can guarantee with all the case studies showing that the average titer can go up to 5 grams per liter and above. The second advantage is we can get a higher correct pairing. We can guarantee 80% above. So with this platform, we can solve these 2 headaches for the bispecific antibodies. Why we can do that? Why we can do the high correct pairing? Because we have we designed a very specific vectors. We can put 4 reading frames into the 1 vector. I think yesterday we are talking about different chains position, up and back and front can affect the quality. So we can make a different kind of the combination of position of each chain to make a different quality of the product. In the 1 vector, we have 4 reading frames. So this is one of the example. We use a 4-chain bispecific as a case study. As you can see, one is for SDS page showing the correct pairing, and the bottom one is showing is the using SES, both showing that is a same correct pairing. Second one, the second slide this slide is just showing that different combination of the heavy chain and the light chain give you different titer. Some is a good titer, like a 6 grams per liter. Some is only 3 grams per liter. So that data just showing us once you have different composition of the vectors can give you different results. Since, just like I mentioned earlier, since we can evaluate our product quality in the cell pool stage, so you can make all kinds of different composition. Then you can make a decision in the early stage. Instead of like a traditional process, you have to make individual cell line, then find the quality. That gives us a lot of advantage, time and manpower-wise, to get good quality. This is the first advantage for the biospecific. Second one is faster. As you can see, we have added is a 1.5 months to get a single colony. So it's a short process. Since the hotspot we identify the hotspot and label with the green protein. So our host cell is a green color. Then we use a homologous transfection process to put our GOI into these cells and to replace the GFP. So the fluorescent cells become non-fluorescent. So in the beginning, the second step, you have a mixture of a fluorescent one and a non-fluorescent one. So that's a color differentiation. Then we can use a single cell sorting steps to easily pick those positive cells. Either use a Satina, VIPS, FAX, or other kind of the regular cell sorting. So that we can make a step 3, we can make a 1 or 2 96 wells to make 4 positive cells. So that you have less work and much faster. So that starting from transfection all the way to get monoclone take about 1.5 months. Also, in the meantime, once you get a single cell into the 96 well, also you can collect all those positive cells into the one pool. With this cell pool, you can make materials, then you can evaluate your product quality. Just like a biospecific, if it is a good pairing or bad pairing, is a good titer or is a low titer. So you can make this all evaluation in the early stage. So that's the second one, is faster. The third one, of course, is going to be high titer and also stable. So this slide is just showing that the real results. We have a IgG1 monoclone, we have IgG4 biospecific, fusion protein, and some is from IVD company, is a highly expressed protein. So average titer on our side is about 5 grams per liter. So if you look at the left one, way left one, the blue one, which is 15 grams per liter, is one of a customer. They use our system, they express by themselves in a 500 liter scale cell culture. So right now they are in the process of the IND filing. The right chart is showing that since the site is stable, so we transfect 8 different products, and then we make a 3-month stability evaluation, just like a standard process, showing that these 8 different products has the stability. So this just showing this technical part. We have a fast process. We can solve the bispecific antibody problem. It's high titer, it's stable. I think a lot of people might have a question is regarding the regulatory issues. Number one, since we have a GFP, we have a integrase, the people might ask, do you still have a gene left over? Do you have a protein left over? So we do have a research showing that we use either NGS or PCR, there's no GFP gene left in the final product, or even in the gene of the product. We don't have a integrase gene left over. Also, when we do the stability issue evaluation, the site is also stable. They don't lose. So this can show this our regulatory questions. This is just an example showing that in the left chart, showing that it is the integrase BXB1 gene that's inactive detected in our cell lines. In the right chart is showing that it's GFP gene, there's no leftover. All right, so this is the cell line development by using AI to get the site specific. But it's not good enough. Because if you want to use a new technology to do the revolution movement in the industry, one platform is not enough. So once you have a good cell line, you must have a good media. So the next few slides just very briefly introduce, we have another form by using AI technology to quickly to screen customized good media. We call it non-screening, because every time if you deal with media, you have to take a lot of time to do screening, either commercial one or in-house one. So with our platform, as you know, in the traditional process on the top part, every time you have to screen all those commercial available media to get your product, to get your media. But in the bottom one, we just take about 1 month. Every time once you use your favorite media to do one-time experiment, if it doesn't work or is not satisfying results, you take the supernatant at day 14 or day 15 or day 16, whatever the process is. Take the supernatant and send to us, send to our company. We have a high throughput measurement. Usually we use LC mass to measure the consumption of those components in the media. Then the AI will kick in to tell us what kind of components are short, how much we need to add, what kind of related component need to add extra, so that in the last part is called optimize the supplement. We add this supplement into your current media and do one more experiment. Normally, we can get good results. So in this whole process, there's no screening process anymore. Do one experiment, do one testing, measurement, do one more test. Then we can get a customized media formulation. So this is a very simple process if you are interested. So you do your experiment in-house. By the end of the process, you take the supernatant, usually about 1 ml, 2 ml, and send to us, and Gravy Bio we can quickly, usually take a few weeks, to get the powder of the supplement, and send this supplement back to the customer and do the one more experiment. So the whole process for the customer-wise, for the end-user-wise, for us is a very simple process. This is the summary of our different kind of the case studies showing that some is starting from 4 grams to 7 grams, some is to 6 to 8 grams, some to 3 to 7 grams. So it's different kind of the case studies showing that within our Alpha OPA platform can easily to make a better performance. So this is a downstream alpha cell with a media. Also, we have an upstream, we're using AI technology we call AlphaDex. This platform will be officially launched in the Bio conference in June. So we use AI for the molecule design. So as you know, in the PCC level for the molecule design, you take a long time. You have antigen, inject the animals, you get to screen those antibodies, you have to do the affinity maturation, harmonization, everything takes almost like 1 year. So we have a platform that using the dry lab to replace those wet lab work. Usually take roughly about 2 months to get these whole things done. So right now the mature one is called AlphaDex, which is molecule sequence optimization. So what we do is whatever you have a sequence which you are not satisfied, particularly for the early stage group, we can upload into our platform. We can make a prediction, see if there's a physical chemical property is good. Is the viscosity is good or bad? Do you have a humanization problem? Do you have a non-specific binding problem? All kinds of things. So we can make a first step is prediction, but it's not good enough. Once find out it's not good enough, we also have another steps using AI, trying to modify your sequence or optimize your sequence to make sure you can have a good humanization score, you have a good affinity maturation, you have a good druggability. So the 3-to-1 package to deliver to the end user that this molecule is acceptable for the next step, normally for the CMC. So this data just showing that our platform, by using commercial product, our accuracy for the humanization is 65%. False positive rate is about 26%. One of the key parameters is called binding energy, antibody energy binding together. If its binding energy is lower, usually lower than 18, minus 18, minus 20 is a good one. So we can use these 60 commercial products showing that our platform is 80%, 85% accurate. Okay. This is another one for the druggability, for the precipitation. I think precipitation is a big headache for most of the drug developers. So we can see that the left chart is showing that we use our aggregation score showing that those sequence is a red warning. As you can see, the sample, this is real data, this wet lab, is everything is crowdedness. So in that, for the precipitation prediction, our accuracy, our platform is very accurate. In the right chart is showing that is lower than score is lower than 1, which is easily soluble, is a soluble product. The accuracy is about 79%. So this is an example showing that for precipitation evaluation. This is one of case studies. One of customer, they sent us 13 sequence. If you look at the red chart, the red circle, this is our dry lab platform to showing that some is red, is warning, some is good, and the blue part is a real wet lab work. As you can see, as long as our platform to predict is warning, the wet lab shows is bad performance. So this is one of a case studies showing that we can help customer to give some warning that some is good, some is bad. Okay, so that's the conclusion. We talk about the cell, we talk about the media, we talk about the drug discovery. So this is the whole Gravy Bio's ecosystem. Starting from the one color, the green part, we can use AI to generate antibody sequence with the dry lab work. Then we can use the yellow part is the druggability assessment, and the blue part we can make a cell line and also media development. The whole thing can become like an ecosystem for the drug development work. What's the real value by this ecosystem in the drug development value chain? So as you know, in the regular process, starting from a beginning imaging to the IND filing usually takes 2 years. Roughly about 1 year to get PCC, another year for the CMC filing, plus minus. With our ecosystem, we can shrink this timeline all the way to 1 year to get the IND filing. So we can save at least 30 months to get the IND file starting from imaging. That's the value for the customer, for the end users with our new technology. Okay, so that's what we have today. So basically what our company is provide a service. Either we can make a cell line for you, or we can out-license this technology to your in-house, or we can make a formulation, we can sell formulation to you, or we can make molecule design for you. So any kind of a business model is available and flexible. Thank you very much.